All right. Well, let's move on to the first keynote. The first keynote address is by Dr. Michael Levin, and the theme is Bioelectricity as Interface to Cellular Intelligence toward Definitive Regenerative Medicine of Injury, Cancer, and Aging. So Dr. Levin is a Vannevar Bush Distinguished Professor of Biology at Tufts University and Director of the Allen Discovery Center. With over 430 publications, his research explores the collective intelligence of cells and applies it to regeneration, cancer, and synthetic bioengineering. So, Dr. Levin, please take it from here.
Thank you very much, and hopefully you guys can all see my slides. I'm going to talk about bioelectricity in a slightly different way than I usually do, and introduce it as a kind of bridge between physics and cognition. And if you want to see the details of all this stuff here, everything is at this website, and this is my personal blog about what I think some of these things mean. I want to point out something that is often forgotten. When you wake up in the morning, each of us may have very abstract, high-level goals. You may have financial goals, social goals, research goals, whatever. And in order for you to get up out of bed and execute on those goals, charged molecules like calcium, potassium, and so on have to cross your muscle membranes. In other words, the chemistry has to dance to the tune of a very high-level, abstract kind of mental goal. How does that happen? How is the chemistry of your body, the actual molecules of your body, controlled by your thoughts and your intentions? This is not some sort of special mind-matter interaction that is only accessible to yoga practitioners or some sort of special masters. This is everyday voluntary motion. This is the amazing miracle of everyday voluntary motion. And our bodies are constructed in a way that can transduce these extremely abstract mental objects like goals and so on, down all the way down to the act of chemistry changing in your cells. And so you might want to ask, okay, this interaction between the mental and the physical, where does it happen? Okay, where is the interface between cognition, right, and the contents of our mind, and so-called just physics or physiology or chemistry? You know, where and how do our thoughts actually affect the physical world? When does the mental become the physical, right? When does it turn over? And what I'm going to suggest is the answer is never. It's cognition all the way down, all the way from high-level minds embodied in human bodies and whatever is beyond that, all the way down to molecules. That's the answer to this puzzle of the interaction of the mental world and physical world. It is mental all the way down. Now, the goal here, of course, is to understand how this happens through the different layers of our body, how to understand communication, the scaling of goals, the remapping of memories across substrates, across the different components of our body, and across the different problem spaces that they solve. And in order to explain what I mean, I would like to introduce you to a single cell. This is an organism known as a lacrimaria, but you get the idea. It's a single cell, and this is what we are made of. We are made of these kind of agential materials. We're not made of passive matter or even active or computational matter. We're made of agential materials, that is, materials with an agenda, materials with competencies. And so there's no brain here. There's no nervous system. You can see this incredible creature and the competency that it has in its environment. And one might wonder, how is it that we could engineer with materials like this? It's completely different than how we used to do it with wood and metal and Legos and so on, because you can't micromanage this kind of thing. You have to convince it to do things. And that's because all throughout the history of life, biology has been solving problems in many, many different spaces besides the three-dimensional space of which we are so familiar. So we can recognize birds and primates and maybe an octopus or a whale, things like that, as intelligent. But long before we had brains, long before we had muscles that allow us to move in three-dimensional space, biology was navigating the space of possible gene expressions, the space of physiological states, and the space of anatomical states. In other words, your journey from being a single cell egg, a fertilized egg, to being a human is a navigation of the space of anatomical possibilities. There's probably an infinite number of possibilities, and these systems do this kind of perception action loop in all of these spaces, but these are very hard for us to recognize. We as humans are completely obsessed with three-dimensional space. If it's not moving around, medium-sized object at medium speeds, we really find it very difficult to understand the intelligence. But in our body, every layer, all the way from the molecular networks to the organelles within our cells, to the networks of cells and tissues and organs, and even collections of organisms, every layer has its own capacity to solve problems. This is the so-called multi-scale competency architecture. At every level of organization, there are agendas and competencies, and they solve problems. I'm going to show you a couple of examples of the kinds of things they do. First of all, this is an amazing creature called an axolotl, and these amphibians are highly regenerative. They replace damaged limbs, eyes, jaws, all sorts of organs. And what happens here is that if the limb is amputated anywhere along the length here, the cells spring into action and they will immediately regenerate and regrow the same pattern, and then they stop. That's the most amazing part about this, is they know when to stop. When do they stop? They stop when the correct axolotl limb has been completed. So it has the ability to know when it's been deviated from its goal, and then to work really hard to get back to that same position in anatomical space. It's the ability to reach the same goal from different starting positions, right? That's pretty impressive. But notice that it's not about damage, and you can see this in the following experiment. In the 1950s, they took tails like this from this amphibian and grafted it, surgically grafted it to the side. And what you see is that over time, that tail transforms into a limb. Okay. Now take the perspective of these these cells here at the tip of the tail. There's nothing locally wrong. There's no injury, no damage. These cells are perfectly correct tail tip cells sitting at the end of a tail. Why do they turn into fingers? Because what's happening here is that a large-scale plan, so the collective, not the actual animal, it's not the brain of the animal, but the tissue and the organ structures are well aware that you don't need a tail here, you actually need a limb, and their high-level intent to fix that error filters down to the molecular biology needed to turn tail tip cells into fingers. It's exactly what I told you at the beginning. It's a multi-scale system where high-level goals of the top-level collective filter down to make the molecular biology and the chemistry adjust to what needs to happen. So the local order actually obeys a global plan. It's an amazing part of our architecture. And so we want to try to understand what are the possibilities here for medicine and for engineering and so on. And we need to start to think about how the outcomes, how the goals of this system, I mean, it's very clearly a goal-seeking system. It repairs, it can remodel. It's a goal-seeking system. Where do the goals come from? So all of us start life as a little ball of blastomeres. And eventually here, this is a cross-section through a human torso. You see this incredible order. Everything is in the right place, the right size relative to the right thing, next to the right structure. Where did this come from? You might think it came from the DNA, but we can read genomes now, and none of this is actually encoded directly in the DNA. The DNA specifies proteins, tiny molecular hardware that every cell gets to have. The question of what kind of symmetry you will have, how many eyes, if you're going to have eyes, how many fingers, all of this stuff is not directly in the DNA at all, any more than the structure of this incredible termite mound or the spider web is in the DNA of those creatures. It is the result of the physiological software that runs on the genetically specified hardware. Now, we have begun to develop tools to decode some of this software. And you might wonder, how do groups of cells remember what to build? How do they know what to build? And what else could you ask them to build? Well, in the brain, we kind of have an idea of how this works. So the brain consists of many cells which have these little ion channels on them. The ion channels allow the cells to have a voltage gradient, and that voltage is communicated or not through these special electrical synapses. And you can see here, this is an amazing video that this group made of zebrafish brains thinking about whatever it is that zebrafish think about. So you can track it and you can try to decode it. So this is the idea of neural decoding, to try to understand what are the memories, the goals, what is encoded in this electrophysiology. Well, it turns out that the ability to store mental content in electrical networks and propagate it down to activity is extremely ancient. Biology discovered this long before we had brains or muscles or nerves. Every cell in your body has these ion channels, so every cell creates these electrical gradients. Most cells have these electrical synapses to their neighbors so they can communicate with them and create networks with large computational capacity. And we can do something very similar to what neuroscientists try to do in the brain, and that is to decode. So here's an early frog embryo, and the colors indicate the voltages that are basically mediating the little conversations that all the cells are having with each other. Who's going to be left, who's going to be right, who's going to be the head, the tail, and so on. And we try to decode this. Can we ask, what does your body think about before you have a brain? And what did bodies think about in general before there were any brains? So we created some tools to do this, to read and write the memories of the body. First, these kind of voltage-sensitive fluorescent dyes. So this is a technology to non-invasively read the electrical communication between cells. And you can see that here in individual cells, and this is an entire embryo. We also do a lot of computational modeling to try to understand quantitatively how this works. I'm going to show you an example of several kinds of patterns. First of all, we call this the electric face. So here's an early frog embryo in a time lapse. The brightness indicates voltage, and you can see here there's a lot going on, but this is one frame of that video. You can see that already long before the organs of the face were specified, the electrical pattern set the future as a scaffold. So you can see here's where the eye is going to be, here's where the mouth is going to be, here are some placoid structures outside. This is how it knows what a correct face looks like. We are literally here looking at the decoded electrical memory of the tissue of what a proper tadpole face is supposed to look like. And it's not just for single embryos, multiple embryos. So each one of these big circles is a different embryo. They communicate with each other. So the cells communicate with each other to make a proper embryo. But the embryos, you can see when I poke this guy here, there's this calcium wave that goes. The embryos communicate with each other to help each other reach the correct endpoint, right? And here you can see we can track this among the individual cells. Now, it's not enough to just read those electrical networks and read the memories that the tissue has. We want to be able to rewrite those memories. We don't use any electric fields. There are no magnets, no electromagnetism, no frequencies, no waves. That is not how we do this. What we do is hack the normal interface that the cells are using to control each other's behavior. And that means controlling these ion channels and controlling the electrical synapses. And we can do this with all the tools of neuroscience. So we can do it with light, we can do it with special pharmacological agents or drugs, and so on. And so now I want to show you just one or two examples of what happens when we can rewrite these pattern memories. Well, the first thing we can do is we can ask the tissue to make whole organs. So here's a side view of a tadpole. So here's the brain up here, the mouth is back here, here's the eye, here's the gut, and the tail is sort of back here. What we've done is inject some ion channel RNA. So these are molecules encoding a specific electrical component called a potassium channel. We inject these cells to set up a little spot of voltage that says build an eye here. And sure enough, the cells do exactly that. Here, they've built this nice eye. It has all the same lens and retina, optic nerve, all the same stuff that an eye is supposed to have. In fact, if we only inject a few cells, so the blue cells are the ones we injected, they will automatically talk to their neighbors and instruct them to join them in making this structure. So all this stuff out here that isn't blue, we never touched it. It's the blue cells tell the other cells what to do. So this is remarkable for many reasons. First of all, it tells us that the bioelectric state, which is what we've manipulated here, in fact is instructive. It tells the tissues what to do. But more importantly, it's instructive at a very high level. We didn't have to tell it what genes to turn on and off. This is nothing to do with genetic editing. This is not about controlling specific genes. This is not about controlling stem cells. We have no idea how to micromanage the creation of an eye. What we do know how to do is to say to the very competent tissue, build an eye here, and it takes care of the rest. That same theme that I started with at the beginning of very high level abstract goals being transduced down into the chemistry that actually implements it. And so all of these things happen after we've said to these cells, instead of a gut, you should make an eye. What we can also do, here's another example, and we have many, many examples that I could show you, but for reasons of time, I'm only going to show you two. This is a flatworm called a planarian. There's a Japanese version called Dugisia japonica. This is a different one called Doratocephala. They have a triangular head like this, and they're highly regenerative. So if we amputate the head, they grow it back. Okay, so they grow back every part of their body, and we're studying this for many reasons. But one of the amazing things is that they use an electrical gradient to know how many heads and what kind of head they should have. And when we manipulate it, we can make them have their normal heads like this, but they can also make flat heads like a completely different species of planarian, or round heads like a different species of planarian. These are between 100 and 150 million years distance from this, but without any genetic change. Okay, so we don't touch the genome at all. We simply communicate electrically with the cells, and we can get the same hardware, so the same genetics, to visit these kind of attractors in anatomical space that belong to these other species. So, and in fact, not just the shape of the head, but the shape of the brain and the distribution of stem cells here becomes just like these other species. So you can ask the body to produce a new organ, okay, in a different location. You can ask the body to produce structures that belong to an entirely different species here. And so this is the beginning of using this bioelectric interface for regenerative medicine applications. And we're working on limb regeneration and repairing birth defects and reprogramming tumors, all of these kinds of things. When we learn the bioelectric interface by which we can communicate with these systems. But notice that everything I've shown you so far has been normal structures that have evolved on Earth. So that eye was a normal frog eye. These heads belong to normal species. And but we need to think beyond this. What is going to happen when, first of all, humans are modified, which we are already modifying ourselves in both in biological and technological ways. So there's going to be an incredible variety of beings that are combinations of novel technologies with existing biological structures. And so we can ask, where do those goals come from? We know where the frog eye came from in the planarian head. They came from evolution acting over eons to set particular structures. But for beings like this that have never been here before, where do the goals of their tissues come from? And in order to try to understand, I want to introduce you to a synthetic life form. So this is something that if you saw, you might think that we got this from the bottom of a lake somewhere, a primitive organism. But actually, if you were to sequence it, you would find 100% Homo sapiens. We call these anthrobots. They are made from human adult cells, not embryonic, adult human patient cells from the tracheal epithelial biopsies. And they have the ability to self-assemble into a tiny little creature that swims around. It has a completely different behavior. It doesn't look anything like any stage of human development. So the plasticity is amazing. When you free your cells from the influence of the rest of the body, they have a different life. And you can ask, what does this thing has never existed before? These anthrobots have no evolutionary history. What do they know how to do? Well, one thing they know how to do is to repair damage, for example, in neurons. So if you plate a bunch of human neurons here in a petri dish, you take a scalpel and you make a big scratch, so a big wound through like this, right? Here's your neural wound. And you put a bunch of these anthrobots labeled in green. They will find a spot in this wound. They will land there. And what you'll see is that they start to knit the neurons across the gap. They start to repair. If you lift them up 4 days later, this is what they're doing. They're repairing the damage. So who would have thought that your tracheal epithelial cells, which sit there quietly for decades dealing with mucus and air particles and so on, have the ability to become a completely different little creature? And so, this is what they look like. They have little cilia, little hairs on their outer surface that allow them to swim. They have 4 different behavior patterns that they can switch between. They have 9,000 differently expressed genes. Okay, so about half the genome is differentially expressed. Again, we didn't do anything we didn't touch the genome. There are no synthetic biology circuits here, no weird nanomaterials, no scaffolds. This is novel capabilities of a being that puts itself together in a way that's never existed in evolution before. And the opportunities here are incredible because these are made from your own cells. You wouldn't need immunosuppression. You wouldn't need to suppress your immune system if these were injected into your body to heal you, right? We're only scratching the surface of what they can do. But they're also teaching us about this amazing scenario where living beings can have goals and properties that were not set by evolution. They don't have an evolutionary history. Where did they come from? Well, we think now that all of these kinds of things, the anthrobots, xenobots, all these other things that we make, are basically vehicles for exploring what you might call the Platonic space, or a latent space of possibilities. These capabilities, these new gene expressions, new behaviors, new forms, new physiological properties, they're not just random things that just happen to be. They are drawn from an ordered, structured space that contains not only mathematical truths, but also high order patterns that we recognize as kinds of minds. And you can see there's a whole symposium that we're running this fall on this Platonic space. This idea that living beings, but also engineered beings like robots and AIs and hybrid forms and cyborgs and so on, are all receive the benefit of the ingressions of patterns from this space. And we really need to understand this to take the full benefit of the technologies that are coming. And so I would suggest that the future of, first of all, let's just talk about biomedicine for a second. These are all the familiar strategies of biomedicine. They're all kind of bottom up. They're all hardware first, but there are all of these incredible technologies coming down the line having to do with exploiting the intelligence of the material of life. And I think that future biomedicine is going to look a lot more like a kind of somatic psychiatry than it's going to look like chemistry. It is really about collaborating, cooperating, and convincing your various intelligences in your body to do the things you need them to do. And the final thing I'll say is this. Besides all the advances that this is going to lead to in regenerative medicine, cancer, birth defects, bioengineering, aside from all that, there's an ethical and a social component here, which is that all of the natural forms that Darwin said when he said endless forms most beautiful are a tiny corner of this enormous space of beings. All of the combinations of evolved material, engineered material, and software, and the patterns from this space, all of these make some kind of a viable, novel, embodied mind. So cyborgs and hybrids and every kind of combination under the sun is coming. We are going to be living with these beings that we already are with. Many of these already exist, but there will certainly be more. And we really need to understand all of these components and develop a novel set of ethical principles to relate to beings that are not like us, that are truly alien, that do not come from our evolutionary life stream. And we need to enter into a kind of ethical symbiosis with these beings as a matter of maturing as a species. So I'll stop here and just thank the students and the postdocs who did some of the work that I showed you today, and our collaborators and our technical support. Disclosure, here are 3 companies that have licensed some of our technologies and support the work. And most of the gratitude goes to the living model systems that have taught us everything we know. So I thank you very much for listening.
Thank you very much. Please give a big hand of applause to Dr. Levin. Thank you so much.